Okay, folks, we're on to the last mini lecture of Chapter 9, and this is I'm going to talk about species distribution modeling and then give you a case study for South Florida for why this matters. So we've talked about how ecology aims to be this predictive science, right? To be able to take some data about something and predict something about the natural world. And one of the really cool modern ways that we do this has to do with population distributions, and these are called species distribution models. And so I'm going to give you a super cursory intro to those. Your book covers them with more detail, and there's as much as you want to learn about these. You could spend the rest of your career working on them. So don't think this is exhaustive, just a primer. So basically, the goal of a species distribution model is to guess where species could occur, right? To guess where populations should persist based on some data, right? And the way this works is we take two pieces of data. We take occurrence data, which are actually the data you're contributing right now, right? Your ecology and action assignments are contributing to data that are occurrence data. We saw this species at this place at this time. And it's basically a map of where individuals are. Some more complicated models also have maps of where individuals aren't. So, for example, if we were trying to detect or trying to decide if habitat was suitable for a skunk, we'd have to go out and see at a random location, is there a skunk there or not? So we would have presences, the presence of an animal, and absences, true absences, they didn't occur there. But for right now, we're just going to talk about presences. So we go out, we find where an animal is on the landscape. And then they have environmental data, typically. And these can be really large data sets, and it's spatial. Really large data sets of sort of temperature and precipitation and incident radiation and any other thing you could imagine, other species co-occurrence, all sorts of crazy things that go into these models. So for every point where we've seen an individual, we have environmental data usually about abiotic, but models can use biotic conditions too, abiotic conditions. Okay? And so you could imagine then that you could take, 1 second, I'm going to pause it, my dog just walked in. Okay, we're back, Charlie's situated. So you could imagine you could take all of these occurrences and say, let's build a model that predicts the best abiotic conditions, the best habitat suitability for a species. And that's explicitly what they do. So you can imagine a map. We're not going to pretend like we draw Florida, but let's draw some land mass here. This is some land mass. We're not even going to pretend like it's real. And we have these occurrence data. So let's put blue dots as the occurrences of our species. And they occur here, and they occur here, and they occur here, and they occur here, right? And then for each of these points, we have a big data frame that has a bunch of abiotic data. So we have temp. We have precip. We have mean annual temp. We have max temp. We have min temp. We have any data you could imagine. And then for each of these points, we have a row, right? We have a row, and we have all these columns. And then what the model does is it says, let's take the best fit parameters here, the best fit abiotic conditions, and then search the landscape for places that it could occur. We'll draw these in purple. So here, oops, that's black. Let's make it purple. Here is a spot maybe that has similar abiotic conditions to where the species occurs in other places. And so maybe it's a good abiotic fit, right? Maybe we expect that if we go out there to find this species, it will occur, right? And that's largely what these species distribution models do. They're spatially explicit, and they try to predict where organisms could occur, right? This is predictive, right? You could say, I have a hypothesis now. I predict that when I go to this peninsula, I'm going to find species X. A really cool example of this is mangroves in Florida, right? So mangroves, red mangroves, a lot of you have seen them. Let's write the name of them here. These are Rhizophora mangle. If you haven't observed one yet, go down to the intercoastal. They're the trees that look like this, with the aerial roots sticking out, and they branch out like this. They sometimes have dangling roots too. And they have a really cool life history, right? They grow in the intertidal. They tolerate salt pretty well, but don't do exceptionally well in high salty environments, but they tolerate salt better than many other plants. And they do a thing that a lot of other mangroves do called live birth, germination, right? So if you look at the mangroves, you'll see a fruit here, this is brown, attached to the adult tree, and the first early leaves of that plant, not technically leaves, but the first early photosynthetic parts of that propagule of the offspring are coming out of that seed, right? The former flower. So this live birth is exceptionally rare in plants. It doesn't happen that often, mostly in mangroves. And what's cool is these seeds fall from trees and then disperse on the currents, right? And Rhizophora mangle is really interesting because it's showing dramatic range shifts that seem to be driven by temperature. And people are building species distribution models to predict where it's going to go. And in fact, it appears as though Rhizophora mangle is moving northward. So let me show you some data that support that. So here's some data from Kavanaugh et al. In PNAS. Let's put this down here. Oh, look, I don't even have to draw a map of Florida. Kavanaugh et al. In PNAS, the Proceedings of the National Academy of Science in 2014. And on this plot, what's really cool is you're seeing on the left-hand side here, black circles represent more mangroves in this long-term study they're doing. So higher density of mangroves over the course of time. And red dots indicate lower density of mangroves over time. And then the size of the dot shows the proportional change. And in some of these places up near St. Augustine, we've seen densities doubling, 100% larger, right? This is pretty wild, right? Up around 29, 30 latitude, 29, 30 latitude, we're seeing 100% change in mangrove density. There's twice as many mangroves per unit area, red mangroves, as there were in the previous sampling time. And part of this is due largely to decreases in hard freezes, right? The climate is changing. These hard freezes that once limited mangrove growth and especially limited the growth of propagules are less frequent. And so we're seeing increased rates. And one of the reasons this is in such a good journal, PNAS is a very good journal, is because they showed this kind of mechanistically. So check out this picture here. These are also data from that same paper. And what you're seeing here is a correlation across a bunch of sites where we're looking at the minimum winter temperature and the delta, the change in percent mangrove area. And you're seeing that in some of these places where it's warmer, so a 0% change means no change. In these places where it's warmer, warmer than 3.6 degrees Celsius in the winter, we're seeing on average increases in growth, right? Only small increases, we're talking like, small percentages. But these add up to lead to doubling of population sizes, doubling of densities over time. And this is pretty spectacular. You can imagine why a species distribution model might not capture this, right? So one of the reasons I picked mangroves when we talked about species distribution models is they have this cool dispersal function, right? Where the propagules germinate on the adults and then float on the water for dispersal. And it turns out that the dispersal of mangroves is largely dependent on currents. And so we might build an SDM, for example, that predicts, we kind of covered up our SDM, but let's pretend that it predicts there should be mangroves here, 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 and here in these pink dots. But the prevailing currents are always running this way, right? If our SDM doesn't account for those currents, then dispersal is what limits the populations from reaching this suitable habitat, right? So SDMs are really great for generating hypotheses about where organisms should occur, where it can be good for them to occur. But then all of the other processes we talked about that limit populations come into play. Dispersal, habitat suitability, the abiotic part is covered by the SDM, but like biotic interactions are largely, they're becoming to be integrated in SDMs, but not super common. And so this is, a perfect capstone. One last really cool piece of data I want to show you is that these same authors, the last author on that PNAS paper, also did a really neat study showing putative evolution of different traits in mangroves at their northern range. So they went out and sampled mangroves from the south end of the range down near Miami and the north end of the range near St. Augustine. and they put all of the plants in a common garden. What this means is they took those propagules and they all put them in the same place to say, let's put them in the same environment and see how they act. And they measured the percentage of plants reproducing. And they found that, you can see clearly here, that plants where they collected propagules from the north end of the range, this expanding range, are much more likely to be reproducing. They invest a lot more in propagules, right? They're spending a lot more of their energy reproducing. And in fact, let me show you another figure. They're investing more in flowering too. So they're investing energy in flowering. You can see source latitude here. This is further north up the coast from St. Augustine, further south of the coast near Miami. These plants from St. Augustine are investing much more in flowers than their counterparts from the south. They're highly reliant on dispersal, right? And this is a good example of natural selection in the wild. This is the front of this species is moving northward. And you can imagine that the mangroves that disperse best are the ones that are moving northward and taking advantage of this newly suitable abiotic habitat where freezing temperatures are disappearing. And because those plants are the best at dispersing, because they reproduce much more highly, because they produce many more propagules, they're dispersing better, and that's driving the range shift. But it's also causing natural selection, right? That dispersal event is the selection event. Only the genotypes that can disperse, only the genotypes that are most successful at dispersing are at the cutting edge of that range expansion. And this is really important for ecosystems in Florida, right? All of these dynamics, mangroves are extraordinarily critical to coastal stability, but they also compete very closely with other coastal habitats, things like sawgrass and seagrass and all these other close competitive interactions, right? And so this northward expansion isn't necessarily a good or a bad thing. It's an ecological change, but we're seeing turnover where ecosystems that were not mangroves historically now are red mangroves because of this change in freezing temperature and evolutionary selection for dispersal. And so I think this is a really wonderful case study to say, okay, we generate a hypothesis with a species distribution model. We predict that, mangroves should be further north than they are because freezes are becoming less frequent. And then we see and test that hypothesis, that's what's happening. We're seeing population density changes. And then we go on to do an experiment. We did an observational thing where we saw populations are changing. Then we do an experiment and show that, the cutting edge, the bleeding edge of this range expansion actually seems to be different, right? Probably via natural selection, via evolution, via natural selection for increased propagule pressure, for flowering and making propagules and dispersing. And so this is a really wonderful case study. It covers what SDMs are. It covers sort of selection. It covers dispersal and habitat suitability, abiotic and biotic habitat suitability. So overall, I think, let's do a quick wrap up here. I'm not going to start a whole new video. But overall, what we've covered in this chapter, right, is 1, populations are group of the same species that could interact with each other potentially, right, and often do. That populations are patchy, and this varies across scales. That ecologists have different ways to count populations, each of which has strengths and benefits, or costs and benefits. Four, that the range of a species, both locally and globally, is driven by habitat suitability. And that's biotic and abiotic, as well as history and dispersal, right? And we talked about this in the case of invasive plant species. We talked about this in the case of mangroves at their cutting edge, right? 5, that metapopulations go through extirpation, that's local extinction, and recolonization. and right, that humans are creating new metapopulation contexts all the time via habitat fragmentation, right? We also covered the types of sort of restoration we might take, thinking about a single large or several small sort of preserves, and that goes into this idea of metapopulation dynamics. Your book covers some math here that I think you should read. You should look at the Levin model of metapopulations. I'm not going to cover it in lecture, but make sure you're reading the chapters. Hopefully by the time you're watching this video, you've already taken the test and you'll see what I mean. I really am going to test you on the book. But that's the key concepts. I don't think I missed anything. Metapopulations, species limited, their distribution variance. Yeah, I think we covered everything. So yeah, enjoy. I'm going to only do chapter 9 this week because I know you have your ecology and action reflections and your test due. That's a big workload for this week. So just watch these lectures. We'll double up at a later week in the semester and catch ourselves up. So have a great week. Good luck on your test. You guys can do it. I believe in you. They're hard, but they're going to be fun. Enjoy. See ya.